For the purpose of this lecture, we will consider the case of small molecule drug discovery in CNS therapeutics. CNS therapeutics stands for Central Nervous System Therapeutics. So these are therapeutics for brain disorders and brain diseases. That could be Alzheimer, Parkinson, brain cancer, including aggressive gliomas and glioblastomas, as well as Parkinson. So let's look at the stages of small molecule drug discovery. At stage 1, you have lead optimization or virtual screening. This is typically done with a computer, but it can also be done by human intuition. This is the development of a scaffold for a drug or therapeutic that fits to a particular target. And we will define those terms in next slides. Once you are happy with that, and that is a very complicated process, you can test the proposed scaffold in an in vitro 2D model. Typically, this is done with a transwell assay. Transwell assay is a sophisticated brain-specific model in which you have a porous brain membrane positioned here, and you can then add your drug into the chamber and see the transport across the membrane in a two-dimensional way. And this allows you to get transport properties, it allows you to get permeability, it allows you to see how the drug would perform in vitro. That is experimental. So we've moved on from the computational or the human intuition optimization stage to an experimental in vitro, and in vitro meaning in the lab. And so there are no animals at this stage, there are no human testing at this stage. We've only done this through a transwell assay. Once we're very satisfied with that result, we can now move to in vivo animal models. This type of model, you can use things like rat brain perfusion, and in this case, the therapeutic is delivered by a cannula into the rat. Upon successful administration of the therapeutic, the rat's brain is then taken out, and one can then estimate the amount of therapeutic that it has transported into the brain. This is a very challenging procedure. It is not routine, but it is also very precise compared to, for instance, the 2D models. There are significant advantages to doing this, but it is also more expensive and has its own set of challenges. And we will not cover this in this course. But this then gives you the in vivo data. And if you're satisfied that there's safety concerns have been looked at and other things, and that the therapeutic is working, you may then seek authorization to go to in vivo human testing. We again will not cover this in this course. But so as you can see from my brief description here, this is a very challenging and very costly hierarchy of testing. Very, very expensive. And the main theme in this is that you have this thing called the blood-brain barrier, BBB. And it seems to be a key limiting factor in CNS drug development. And this is because the BBB has exquisite selectivity. It rejects 98% of molecules from crossing into the brain. And here we have a diagram of the blood-brain barrier. So here we have endothelial cells, brain microvascular endothelial cells. And as you can see, the cells are tightly packed next to each other. So this is one cell, then you have another cell, then you have another cell. And this tight packing it constitutes what's called a tight junction, and it blocks the ability for a therapeutic to cross paracellularly. So that is through the cell-cell spacing. That cell-cell spacing is blocked because of a certain number of proteins and so on, but it's effectively blocked. And so for normal small molecule therapeutic, the main passageway into the brain is via passive diffusion. This passive diffusion is slow and is not possible for all drugs. Furthermore, as the drug crosses in, if it is a target for certain proteins sitting in the membrane called efflux pumps, it will then diffuse over here, bind to the protein, and get pumped out again. This in particular is a challenge for chemotherapeutic agents and other useful therapeutics that are recognized by the cell as cytotoxic. Okay, so this efflux pumping lowers the activity, and we will discuss this in a little bit. For other things like ions and water, you have transporters that send them across. Okay. And so because this process is so expensive and difficult, we would like to simulate the cellular transport across the blood-brain barrier with computers. And the advantages of this is you can estimate the permeability. You can also determine the transport mechanism, and you could replace the first two steps in CNS drug development. And today's lecture will present computational methods for this. Okay, let's look at some early models for CNS drug development. The first model that came from the company Pfizer by a scientist called Chris Lipinski. And he developed what was called Lipinski's rule of 5. And this was a set of parameters that, when obeyed, allows you to predict if a drug will cross the blood-brain barrier. And his suggestion was, well, here are the chemoinformatic parameters for a compound to cross the blood-brain barrier. That a compound should have a molecular weight less than 500 Dalton, or that is 500 grams per mole. It should have a dipole of less than 5 dBi. It should have a lipophilicity of less than 5, so not too hydrophobic. It should have a certain number of hydrogen bond acceptor sites, less than 10, and a certain number of hydrogen bond donor sites, less than 5. These, particularly the last two numbers, means that you don't want too many hydrogen bond donors and acceptors, because then the compound might get stuck in the process of attempting to cross. If it's too hydrophobic, it will get stuck in the membrane. It won't want to leave. And if it's too polar, it won't be able to cross the membrane because the membrane has polar head groups. Okay. This was a pretty reasonable suggestion and proved to be very popular until, people realized that, yeah, it works. These are the compounds for which it works. And this is on a scale of drug likedness. So the closer it is to a canonical drug, yes, it works. But in particular, for low drug likedness, that could be peptides, other things, antibodies, it really fails very critically. So this means that there are drugs that should cross according to Lipinski that don't cross. That would be drugs that have a small molecular weight, but simply don't cross for other reasons. And then there are drugs that shouldn't cross, for instance, large peptides and even large antibodies. They shouldn't cross according to Lipinski's rule because they're too big. However, there are antibodies that cross the blood-brain barrier. So these are therapeutics that violate Lipinski's rule, but nevertheless work. And so it turned out that we can't rely on this rule for all estimation of whether a therapeutic will cross the blood-brain barrier. So then people moved to Bayesian pass-fail models. These models ask the question, will the therapeutic you have developed cross the blood-brain barrier, provided it has a certain number of parameters, values. So this is based on Bayesian statistics. Bayesian statistics in which you ask a question, PA given a B happens. Okay, so you can think of this as, probability that I will get to university on time, given that it rains. Okay, that's Bayesian statistics questions, okay, of that type. So here it's P, the probability that a drug crosses, given that it has a certain partition coefficient, a certain van der Waals surface area, a certain polar surface area, etc. And you can also train this with AI now, and you can also do what's called a Pinsky refinement by adding more parameters to your model. And here are some papers if you're interested in this model, and this has recently been of interest. However, we really would like to move to simulations. So simulations, in silico simulations, provide quantitative, high-resolution, molecule-specific information. So as you can see here, this is a simulation of a drug crossing the blood-brain barrier. You can see the drug as it crosses in time. You know exactly how long it took to cross. You know how long it took to move a certain distance. You know which atoms it interacted with. This is your brain membrane. And so this type of model is really useful because it provides quantitative, high-resolution, molecule-specific information. And so we would like to investigate this type of methods more, and these methods are now gaining a lot of popularity. So in this lecture, we will now go by theme, okay? So first, we will start with combinatorial chemistry. And what is that? So combinatorial chemistry is a lab technique in which millions of molecular constructions can be synthesized and tested for biological activity. Combinatorial chemistry has generated a massive number of targeted molecules for testing. Combinatorial chemistry has led to techniques such as high throughput screening that automate the screening process so larger numbers of biological assays can be done. Effectively, what this tells us is that combinatorial chemistry has sped up the process of drug discovery. And an old estimate suggested it had reduced the discovery to market time from what used to be 10 to 14 years to 5 to 8 years. And this was 20 years ago. So likely this estimate is even faster now. So let's look at a little bit of how combinatorial chemistry compares to very conventional drug discovery methodologies. So in a conventional drug discovery molecule process, you would develop only one molecule at a time. This would be very slow. And you would follow the philosophy of make, purity, test. So you make the compound, you test purity, then you test the activity on the target receptor that you want. Does this, can we make the compound? Can we attain the purity that we need? And does it work on our receptor? At best, in a large company, at best you can do 100 molecules a month, and this therefore means slow lead generation. And a lead compound is the most interesting compound in your database. And this conventional method carries a high risk of failure. Okay. So now let's look at what combinatorial chemistry can add to that. Combinatorial chemistry is associated with a lower risk of failure. It still follows the same philosophy: make, purity, test. So that is, you make the compound, you test the purity that you need, and then you test the activity on the receptor. Combinatorial chemistry allows you to get to thousands of molecules a month at the moment. And it is associated with a faster lead generation and therefore a lower risk of failure. The synergy between those two is called hit to lead, and this is in general the philosophy we will use, hit to lead. And I will explain what this means now. So the hit to lead philosophy within combinatorial chemistry and within the drug design pipeline goes as follows. Step 1, propose a therapeutic target for a disease, for instance. That could be a receptor binding pocket that is known to modulate or affect the disease. Then, using combinatorial chemistry methods, propose or perform a lead compound discovery. So this is step one is here, your target binding site. That's your therapeutic target. Then step two is this lead compound discovery, and that can be done in many ways. One way is called hit identification, that results in a hit to lead. So you start with a scaffold, and then you optimize the scaffold, and that's called hit to lead. You start from something that has low activity, and then you increase the activity by adding things to the scaffold that are favorable to binding to that therapeutic target. That's called hit to lead. Another type of method is called fragment identification. So you look at fragments that might favorably bind to that binding site, and then you connect them in ways that are favorable. This is called fragment to lead. Okay, and this final process is called lead optimization. Okay, so either hit to lead, fragment to lead, you can do it in many ways. When you finally find the most optimal binding candidates, there could be thousands of, hit to lead compounds or fragment to lead, and you look at the activity at that particular binding site, and you develop a candidate. So a candidate with the most efficient binding under the conditions that you need, with the correct purity and all the other things. And that, provided all the other things, and you can do clinical trials, that becomes a drug on the market. And this is a very expensive process. Today, this pipeline is $1 billion for a successful FDA-approved compound on the market. So it is very, very expensive. And we certainly need combinatorial chemistry for this process. Okay. Now, combinatorial chemistry can really impact this step, the therapeutic target identification, by looking genomic analysis and other scans of databases can identify what gene and therefore what protein might be associated with a disease. And then combinatorial chemistry and the production of massive amounts of leads therefore really impact the lead compound discovery, as opposed to starting from just one lead at a time with the conventional methods.